Good morning, everyone. Thank you, Mr. Chair. Thanks for the opportunity to share with you a few thoughts about genomics this morning. I won't go into detail presenting myself. You have my bio in the small booklet, so if you want to read it. But sorry, I'm not as disciplined as the other speakers this morning because I'm coming from an academic background. So I spent 20 years in research, and you know how researchers are, okay? So sorry for that. I will cover three topics shortly this morning. First, to tell you a little bit about genomics and cover some of the application of genomics and then looking into the future and make some conclusion. One very unique thing is that all living things contain DNA. And that's really a very nice characteristic because DNA is actually the encyclopedia of life. It's defining the way you are looking today and the way also you will be looking tomorrow. So that's a very nice characteristic. All living things contain DNA. The second good news for this morning is that each individual has a unique DNA sequence. So if you had the possibility, and there are some ways, some technology available today, if you had the possibility to read your DNA, so your encyclopedia of life contained in each of your cells, you would see that your DNA is different from the DNA from your neighbor. Not too different, but a little bit different. Different enough that you can distinguish between two people. And that's a very nice thing. Why? And I could argue this morning that actually DNA, that's the ultimate identification, the ultimate traceability that you could have. But the bad news is that DNA is not visible. So it doesn't fit with the ICAR standard. So that's a bad thing. And I guess another bad news is that it's quite difficult if you are in the field to compare the DNA sequence of two animals because there is not much you can do in the field, and you want to do it in the field. But the good news is that DNA, and it was already mentioned this morning, is DNA will stay forever in an animal. So you have the same DNA. So you have the same encyclopedia of life. And we are very fortunate in agriculture, in animal and plant species, that some people started some project a few decades ago, to sequence the entire human genome. So to be able to crack the sequence of the DNA, the encyclopedia of life for several organisms. And so the race to sequence the human genome resulted then in a big improvement in technology to sequence, to have access to the DNA, to read it, and this improvement in technology resulted in a 100 million-fold decrease in DNA sequencing prices since 1990. And it's continuing to decrease, so costs are coming down. What costed $3 billion to sequence a human genome a few decades ago, what costed $53 million to sequence a bovine genome doesn't cost any more than $1,000 to $3,000 today. So that's a good news, and so that's why scientists started to sequence genome of animal and plant species, and they resequence in order for what? To discover the small differences between the individual, between the different breeds of animals that you have, but within a breed, the differences that you have within the breed between the animals. And it's clearly a new way of looking of the encyclopedia of life that we are also calling the genome. Sequencing and resequencing animals from the same breed, but different animals or animals from different breeds, led to the discovery of millions of genetic markers, so no more microsatellite, but what we call single nucleotide polymorphism. Why that? Because they are based on just a single base pair differences on the DNA molecule. And that's very powerful because if you have those markers, you can start tracking the DNA, so the chromosome or the chromosome pieces from one generation to the other. And even if the marker is not in a region coding for a gene, so a cooking recipe, if they are just in vicinity of those, then they can track for the variability nearby. So they can track for some characteristic that are on the DNA molecule nearby. And that's very powerful. So that's clearly a new way of looking into the genome. So an improvement in sequencing technology, so getting cheaper. Another big technology breakthrough was the fact that those genetic markers, we were able to develop technology to deploy them massively in parallel fashion on a DNA microarray. And each of you receive in your bag a small package with a DNA microarray, where you have on this array the equivalent of 777,000 SNPs for 8 different samples that could be genotyped with. Just how powerful the system could be. And that's another picture of a microarray. So what such a microarray really enabled today, that's to look at the genome, the encyclopedia of life, in a complete different way. And start looking what is transmitted from the ancestor to the offspring, and being able to trace the different chromosomes, how the segregation is going on, how the crossing over are going on from one generation to the other, and starting to make even some choices based on what you are reading with those genetic markers. And so it's clearly a new way to look and to discover how the phenotype is clearly the expression of the genotype and the environment. That's completely new. And there are some very big expectations from genomics. If you have access to the genome information, if you can track variation on the genome level, then you can better understand the genome structure, the function, how it works. And then you can make some work and discovery about the genetic control for complex phenotypes and having some biomedical models and so on. For the industry, on the commercial side, it's really a new way to increase efficiency of animal breeding programs. It's also a new way of getting a better insight into reproduction growth, nutrition, health, animal welfare, increased quality of, and security of animal products. So clearly genomics is seen today as being able to provide the tools needed to refine and more rapidly achieve breeding goals. I want now to move on and describe just a few studies, a few genomic applications that were done using either sequencing or using those DNA microarrays, the chips. Here, a work that was done from one of my previous PhD when I was in research. We were looking into trying to rebuild the genetic history of some French cattle. And so we sampled 47 populations from different parts of the world. We had a total of a little over 1,000 animals that we genotype with DNA chip with 54,000 SNPs, so 54,000 genetic markers. And we got 44 very good quality ones. And then we tried to study, can we reconstruct, as those animals are coming from different breeds, different populations, can we reconstruct who is coming from where or from what? And that's the tree that is showing that using those markers, we were able to reconstruct the different breeds that we had in our sampling population in the lab. And we were able even to distinguish, like you see, West African taurine, Zebu Indian origin, and the European taurine here. So you could even go and do the same with crossbred animals and see the part of the genome that is coming from one breed or the other. So very powerful analysis that you can do. Another possibility of using genomics today is to do it on a population level and trying to look to what is called signature of selection. You can also in population, in your population, also if they are out in the field and you don't think that they are selected, you could have some natural selection going on. And so the goal is trying to use DNA markers to identify targets of selection, either natural or artificial. Quite sure what genomics can provide today is also parentage and traceability. And you are here also dealing with a few genetic markers that are recommended by the International Society of Animal Genetics. They put together a list of 200 SNPs that can be used in bovine parentage, and the labs that are running usually the parentage verification, they are well organized, they are running comparison tests, and they are also getting an accreditation by ICAR. So it's all a process that can be done today with genomics. Another application of genomics is finding genes of interest. Just one study that just came out last December, I think, about the Senepal breed of cattle, where they discover a dominant genetic syndrome, and that was rapidly propagating through their population, and because of the use of one bull and his son through artificial insemination. And they had an unusual phenotype that they were discovering in their population. They had a phenotype with lactation failure, excessively hairy pelage, and thermoregulatory dysfunction. I don't want to go into all the detail of that study, but just to show you the wild type, the normal Senepal, and the mutant, you can clearly see the difference. So what they did is, again, here, they were taking a little over 100 animals, and they did a comparison of those animals and a whole genome association study to look, by using 100,000 of genetic markers, can we discover a region of the genome that is strongly associated with the phenotype that we are observing in the population. And here again, the result on all the chromosomes, 1 to 29 should be also there. But and if you have a peak, that means that you have an effect. You have some SNPs associated with the phenotype, and you are clearly seeing on chromosome 23, there is a big peak. And so the gene was mapped. It was a prolactin. They found the causal mutation. And in addition to that, by sequencing, they found a second gene, the prolactin receptor, that was also responsible for the other part of the phenotype. So very powerful study that you can do, but one condition is that you get the right phenotype. You get the right observation, and you're able to sample your animals. A last point I want to make is about genomic selection. Having access to all those genomics markers, people are now ready to use the marker data to predict the performance of the animals. So being able to predict the genetic value of the animal. There are a lot of papers describing that. It's really a straightforward process where you need to be able to start with a population for which you have phenotypes, so you have performance recording, and you have biological samples so that you can genotype, and then you can genotype all those animals and evaluate the effect of each of the markers that you have on the specific performances that you are recording. And so, based on that, on the different effects that you are getting, you can compute a prediction equation that you can then use in a population for which you have no recording in place, and you don't need to have it, but you can genotype the animal, you can compute their breeding value, and you can start ranking those and select the best for further use. So that's clearly the path towards genomic selection. It's clearly a paradigm change for breeding, as in addition to the pedigree and phenotypic information, you are adding the genomic information to that. That means that if you have access to the genomic information, that means that very early in age, you can do a genomic prediction as soon as you have access to DNA. It could even be done on embryo or right after birth. And you can not only select on the male side, but you can select also on the female side, so both genders. Now, one of the problems is that, okay, it's good for the developed country because they have been recording phenotypes and pedigree for more than 100 years. But how do we work now? And they have been implementing progeny testing for nearly 50 years. So what about developing countries? Usually there is a lack of programs that record phenotypes on pedigrees animals. There is a lack of evaluation of our national testing program to assess the genetic value of germplasm. But I guess that's why you are here all together. It's to start the process or to improve the process that is starting or that is getting in place in different countries. But it shouldn't retain you to start using genomics, because you can start using genomics to identify critical populations for preservation or to identify well-adapted local breeds that could be utilized and in combination with selection and crossbreeding in the future. So if I try to sum up, just a few applications of genomics that I very briefly shared with you is clearly genomics. The basis could be done on parentage verification, traceability, that's clear. I show you one example or two about population genomics. It can really help knowing a little better the structure of the population, characterizing the animal genetic resources, and identifying signature of selection that could be worth looking in the future. A big promise of genomics is all about genomic breeding, where you can improve animal production, so it's really genetic improvement, but while you can also conserve the indigenous animal genetic resources. Genomic selection is clearly one of the big applications, but for that, I want to state it again, you need recording in place and you need to be able to sample animals. One topic that I didn't cover, I don't have the time, sorry, but animal health, genomics can also help in pathogen identification, in pathogen characterization, and also setting up some very powerful diagnostic. Critical point, and I think that's my last slide, critical point to consider for genomics, it's not a magic tool. It's clearly not a magic tool. There is no genomic without pertinent and accurate phenotypes. So that's the basis. Really, recording and recording well is the basis. It's the trash in, trash out principle applies also for genomics. So that's, you need good recording. Otherwise, you won't be able to do a good job with genomic. Don't think that genomic will replace proper management. No, not at all. It can help if all the other steps are in place, but first you need to put in place all the recording and all the sampling. If I speak very briefly about genomic selection, I would say it's impossible today to predict the weather with 100% accuracy, even with all the scientific improvement that have been done over the past decades. And likewise, genomic selection is not an exact science today, but it's a powerful new tool for predicting results. And Larry Schaefer from CGIL said the potential advantages of a genome-wide selection scheme are too great to ignore. And I would say also, with all the genomic application, yes, it's too great to be ignored today. So let's start doing genomics today. And I think I will finish here because my time is over, but here on this slide, I'm making a few predictions for the future about that genomic selection will become really holistic in all the regions of the world, and that clearly sequencing will become also a commodity in a few years from now. Thank you for your attention.